Sustained focus is fundamentally constrained by the brain's consumption of energy and its finite cognitive resources. The prefrontal cortex, which governs executive functions such as decision-making, working memory, planning, and self-control, is particularly metabolically demanding. As this region remains highly active during periods of concentrated work, it requires substantial glucose and oxygen to maintain neuronal firing rates and efficient synaptic communication. Over time, the continued engagement of these circuits leads to gradual depletion of energy substrates, causing neural fatigue that manifests as reduced cognitive efficiency, slower processing speed, and lapses in attention. The interplay between the prefrontal cortex and subcortical arousal systems, including the locus coeruleus and basal forebrain, further highlights the limitations of sustained focus. These subcortical regions regulate alertness through neurotransmitters such as norepinephrine and dopamine, which modulate arousal and the brain's responsiveness to stimuli. As these neurotransmitters fluctuate or deplete during extended periods of concentrated effort, attentional networks struggle to maintain optimal function, resulting in mental fatigue, decreased motivation, and errors in task performance. The depletion of cognitive resources is compounded by the natural buildup of adenosine, a neuromodulator that signals the need for rest and sleep. During extended mental activity, adenosine accumulates in the brain, particularly in areas involved in attention and executive function, creating an internal pressure that makes sustaining focus increasingly difficult. This mechanism acts as a protective system, ensuring that the brain does not overextend its metabolic capacity, but it also sets a natural limit on how long intense focus can be maintained. The balance between energy availability, neurotransmitter regulation, and the accumulation of fatigue signals defines the window in which peak cognitive performance is achievable. Ignoring these physiological constraints by attempting to work beyond them often leads to diminishing returns, as the quality of attention and the efficiency of cognitive processes progressively decline. Neurochemical changes during sustained focus further illustrate the limitations of prolonged work. Dopamine, which supports motivation, reward, and goal-directed behavior, can decrease with extended cognitive effort, reducing the perceived value of continued engagement in a task. Norepinephrine, critical for maintaining alertness and attentional focus, can also fluctuate, leading to lapses in vigilance and increased distractibility. These fluctuations are not uniform. Individual differences in receptor sensitivity, baseline arousal, and metabolic efficiency influence how quickly fatigue sets in. Consequently, some individuals may maintain high levels of focus for longer periods, while others experience rapid declines in cognitive performance. Understanding these neurochemical dynamics underscores the importance of aligning work intervals with the brain's natural capacity for sustained attention. Attention itself is not a monolithic resource, but a dynamic system that shifts across networks in the brain. Focused work relies heavily on the central executive network, which allocates cognitive resources, suppresses distractions, and maintains task-relevant information. Over time, prolonged engagement in a single task fatigues this network, leading to increased susceptibility to mind-wandering and reduced working memory function. Simultaneously, the default mode network, typically suppressed during active task engagement, may become more active, signaling a drift toward internal thoughts and reduced external focus. These neural shifts reflect the inherent limits of attentional control and the need for strategic breaks to restore optimal function. Environmental factors, including distractions, noise, and cognitive load, exacerbate the depletion of attentional resources. High-demand environments increase metabolic consumption and accelerate the onset of mental fatigue, whereas optimized conditions can extend the duration of effective focus. Physiological states such as hunger, dehydration, sleep deprivation, or elevated stress levels further modulate the brain's capacity to sustain attention, influencing both subjective experience of fatigue and objective performance metrics. Recognizing that sustained focus is constrained by a combination of metabolic, neurochemical, and environmental factors allows for strategic planning of work sessions to maximize efficiency and minimize cognitive depletion. By understanding that the prefrontal cortex and subcortical arousal systems operate within finite energy and neurotransmitter limits, individuals can optimize the duration and structure of focused work sessions, ensuring that attention, motivation, and cognitive performance are maintained at their highest possible levels. The concept of optimal focus intervals is rooted in the natural rhythms of attention, arousal, and cognitive energy. Human cognitive systems are designed to operate in bursts of high engagement rather than in prolonged, uninterrupted states of concentration. Research suggests that the brain can sustain peak attention and performance for approximately 60 to 90 minutes before the efficiency of neural circuits begins to decline. Within this interval, the prefrontal cortex and associated attentional networks maintain heightened activity, supporting executive functions such as decision-making, working memory, problem-solving, and goal-directed behavior. During this period, neurotransmitters such as dopamine and norepinephrine remain sufficiently elevated to sustain motivation, alertness, and cognitive efficiency, allowing individuals to tackle demanding tasks effectively. The decline in performance after extended focus is closely tied to the depletion of both energy resources and neurochemical support. Continuous activation of the prefrontal cortex consumes glucose and oxygen, which are required for sustained neural firing and synaptic transmission. As energy substrates diminish, neuronal signaling becomes less efficient, leading to slower processing speeds, reduced accuracy, and lapses in attention. Concurrently, the levels of key neurotransmitters such as dopamine, which supports motivation and reward, and norepinephrine, which enhances alertness and vigilance, can fluctuate or decrease, diminishing the ability to sustain engagement. These physiological limitations highlight why attempting to maintain focus for excessively long periods often results in diminishing returns, as cognitive performance no longer matches the initial level of efficiency. The 60 to 90-minute focus interval aligns with findings from studies on ultradian rhythms, which are natural cycles of physiological and neural activity that occur throughout the day. These rhythms influence arousal, attention, and mental energy, creating predictable peaks and troughs in cognitive performance. By scheduling work sessions to coincide with these natural peaks, individuals can take advantage of periods when attentional networks and arousal systems are most receptive, maximizing efficiency and reducing mental fatigue. Ignoring these rhythms and attempting to extend focus beyond natural limits can accelerate cognitive depletion, impair decision-making, and increase susceptibility to distractions. Task complexity also interacts with optimal focus intervals. Tasks that demand high levels of executive control, creativity, or analytical thinking require more frequent replenishment of neural and metabolic resources. For these tasks, shorter focused sessions within the 60 to 90-minute window, followed by strategic breaks, can help maintain consistent performance. In contrast, less demanding or repetitive tasks may allow individuals to sustain attention for slightly longer periods without significant decline, as the cognitive load and metabolic demands are lower. Tailoring the duration of focused work to the complexity of the task ensures that cognitive resources are efficiently allocated and that performance remains high. Individual variability further influences optimal work intervals. Genetics, baseline arousal, prior sleep quality, and overall cognitive stamina determine how long a person can maintain peak focus. Some individuals may find that 50-minute intervals are sufficient to sustain productivity, while others can maintain high performance closer to the 90-minute mark. Monitoring subjective indicators such as perceived effort, fatigue, and engagement, along with objective measures like error rates or reaction time, allows for personalized adjustment of focus intervals. This self-monitoring helps individuals optimize work duration according to their neurocognitive profile, balancing effort and recovery. Breaks play a complementary role in maximizing focus intervals. After 60 to 90 minutes of concentrated work, even a brief pause of 5 to 15 minutes helps restore neurotransmitter balance, replenish glucose in the brain, and reset attentional networks. These breaks prevent cognitive overload, reduce mental fatigue, and enhance the ability to return to high-level focus with renewed efficiency. Incorporating breaks into structured work intervals leverages the natural rhythm of attention, allowing individuals to sustain productivity across multiple sessions without sacrificing performance. By aligning work intervals with neural and metabolic constraints, individuals can optimize attention, motivation, and cognitive efficiency, ensuring that each period of focused work produces maximal output without accelerating fatigue. Strategic timing of work and recovery allows the brain to operate within its natural capacity, enhancing both performance and sustainability of effort. Breaks are a critical component of maintaining high levels of cognitive performance and sustained focus. The brain's attentional networks and prefrontal cortex, which are responsible for executive functions such as working memory, decision-making, and inhibitory control, are metabolically demanding and require periodic recovery to maintain efficiency. Even short periods of uninterrupted focus lead to gradual depletion of energy substrates, including glucose and oxygen, as well as fluctuations in key neurotransmitters such as dopamine and norepinephrine. Without recovery intervals, these physiological constraints manifest as mental fatigue, reduced accuracy, slower processing speeds, and increased susceptibility to distraction. Breaks provide an opportunity for the brain to restore these essential resources, allowing neural circuits to recover and resume optimal performance in subsequent work periods. The timing and duration of breaks play a pivotal role in their effectiveness. Research suggests that brief breaks lasting between 5 and 15 minutes are sufficient to restore attentional capacity without causing a loss of task momentum. These short intervals allow the prefrontal cortex to downregulate high-demand neural activity, while subcortical arousal systems recalibrate. During this time, neurotransmitter levels stabilize, mental effort decreases, and subjective feelings of fatigue are reduced. Even brief shifts in attention, such as standing, stretching, or engaging in light sensory stimulation, can enhance recovery by promoting blood flow, increasing oxygen delivery to neurons, and resetting neural oscillations associated with attention. The goal of these breaks is not to disengage completely, but to provide a restorative pause that prevents the cumulative effects of fatigue from impairing performance. Breaks also facilitate cognitive consolidation and memory retention. During periods of focused work, the hippocampus and related structures actively encode and consolidate information. Pausing allows these processes to continue efficiently by reducing interference from continuous cognitive load. Brief periods of rest promote neural replay and strengthening of synaptic connections formed during the preceding work interval. This mechanism is particularly relevant for learning tasks, problem-solving, and creative endeavors, where memory consolidation and insight generation are essential. Strategic breaks therefore support not only attentional restoration, but also the quality and durability of cognitive outputs. The type of activity during breaks further influences their restorative effect. Engaging in light physical movements such as walking or stretching enhances cerebral blood flow, oxygen delivery, and arousal regulation, which supports renewed focus upon returning to the task. Mindfulness or brief meditation can reduce stress and mental tension, resetting neural networks involved in attention and emotional regulation. Even brief exposure to natural environments or changing sensory input can provide cognitive relief by reducing mental fatigue and preventing attentional depletion. The key is to select activities that promote recovery without introducing new cognitive demands that could offset the restorative benefits of the break. Individual differences affect both the frequency and duration of optimal breaks. Factors such as baseline attentional stamina, circadian rhythms, prior sleep quality, and genetic variations in neurotransmitter systems determine how quickly mental fatigue accumulates and how effectively recovery occurs. Some individuals may benefit from slightly longer breaks to restore cognitive energy fully, while others may require shorter, more frequent pauses to maintain consistent focus. Monitoring subjective cues, including perceived mental effort, fatigue, or irritability, can guide the timing of breaks, ensuring that recovery is personalized and maximally effective. Environmental context also modulates the benefits of breaks. High-demand, distracting, or stressful work environments accelerate cognitive depletion, making restorative breaks more necessary. Conversely, optimized conditions, quiet spaces, natural light, ergonomic seating, and minimal interruptions can extend the duration of effective focus before a break is needed. Combining strategic breaks with an optimized environment enhances both the efficiency and sustainability of concentrated work. By incorporating well-timed breaks into structured work intervals, individuals can restore neurotransmitter balance, replenish metabolic resources, and recalibrate attentional networks. Breaks prevent cognitive overload, sustain motivation, and improve both the accuracy and efficiency of subsequent focus periods, making them an essential tool for maintaining high-level performance over extended work sessions. Individual differences play a significant role in determining the optimal length of focused work and the capacity to sustain attention over time. While research provides general guidelines, such as 60 to 90 minutes of concentrated effort followed by a break, these intervals are not universally ideal. Genetic factors, baseline arousal levels, prior sleep quality, stress resilience, and habitual attentional patterns all influence how long a person can maintain peak cognitive performance. For example, variations in genes that regulate dopamine and norepinephrine signaling affect the efficiency of the prefrontal cortex and subcortical arousal systems, influencing both the duration and quality of sustained focus. Individuals with more robust dopaminergic signaling may experience higher motivation and better resistance to mental fatigue, allowing slightly longer work intervals before attentional decline occurs. Baseline arousal and circadian timing are also critical determinants of individual focus capacity. Each person has natural peaks and troughs of alertness throughout the day, driven by circadian rhythms and ultradian cycles. Some individuals naturally experience peak cognitive energy in the morning, while others perform best in the afternoon or evening. Aligning work intervals with these periods of heightened arousal allows for more efficient use of attentional resources and maximizes performance. Conversely, attempting demanding cognitive tasks during periods of low arousal increases the likelihood of fatigue, errors, and reduced efficiency, regardless of general guidelines on optimal focus intervals. Sleep quality and overall cognitive stamina further modulate attentional endurance. Chronic sleep deprivation or fragmented sleep reduces prefrontal cortex efficiency, disrupts neurotransmitter balance, and impairs the ability to sustain focused work. In such conditions, shorter intervals with more frequent breaks are often necessary to maintain acceptable performance levels. Conversely, well-rested individuals with high baseline cognitive stamina can extend their focus periods within the general 60 to 90-minute range, demonstrating the importance of personalized scheduling based on internal physiological and cognitive states. Habitual attentional patterns and training also influence focus capacity. Individuals who regularly practice sustained attention, either through work routines, meditation, or cognitive training exercises, tend to build resilience in attentional networks. This attentional endurance allows them to maintain focus for longer durations, with reduced susceptibility to mind wandering and distraction. Conversely, individuals unaccustomed to extended cognitive engagement may experience faster onset of mental fatigue and require shorter work intervals and more frequent restorative breaks. This variability highlights the importance of gradually adapting focus durations to individual capacity rather than strictly adhering to standardized recommendations. Monitoring subjective and objective indicators of cognitive performance is essential for tailoring work intervals. Subjective cues, such as perceived mental effort, irritability, or difficulty maintaining concentration, can signal when attentional resources are approaching depletion. Objective measures, such as error rates, reaction times, or the quality of completed work, provide additional feedback on the effectiveness of a given focus interval. By tracking these indicators, individuals can adjust the length and frequency of work periods to align with their unique neurocognitive profile, optimizing both productivity and sustainability. Environmental and lifestyle factors also interact with individual differences to influence attentional endurance. Nutrition, hydration, stress levels, and physical activity all modulate prefrontal cortex function and arousal systems, affecting how long attention can be sustained. High-stress environments or cognitively demanding conditions accelerate fatigue, while supportive, low-distraction environments extend the duration of effective focus. Likewise, balanced nutrition and regular exercise improve energy metabolism and neurotransmitter regulation, enhancing the brain's capacity to maintain attention. By considering genetic factors, circadian rhythms, sleep quality, habitual attentional patterns, and lifestyle influences, individuals can tailor the length of focused work sessions to their unique capacity. Personalized scheduling ensures that attentional resources are used efficiently, minimizing fatigue and maximizing cognitive performance. Recognizing that there is no one-size-fits-all approach allows individuals to optimize both productivity and sustainability in their work and learning routines. Task complexity is a critical factor in determining the optimal length of focused work and the structure of work sessions. The cognitive demands of a given activity dictate how quickly attentional resources are depleted and how frequently restorative breaks are required. Highly complex or novel tasks such as problem solving, strategic planning, or creative work place significant metabolic and neural demands on the prefrontal cortex and associated attentional networks. These activities require sustained activation of executive functions, including working memory, decision-making, inhibitory control, and error monitoring. Because these neural systems are metabolically costly and sensitive to fatigue, attempting to maintain focus on high-complexity tasks for extended periods without breaks often leads to declines in accuracy, slower processing, and increased cognitive errors. In contrast, routine, repetitive, or less cognitively demanding tasks require lower levels of executive function and prefrontal cortex engagement. Activities such as data entry, basic calculations, or habitual physical tasks can often be sustained for longer periods with minimal decline in performance because the neural systems involved are less vulnerable to rapid depletion. While motivation and arousal still play a role in these tasks, the lower cognitive load allows individuals to maintain attention for longer intervals without experiencing significant fatigue. Recognizing the differences in cognitive demands is essential for structuring work sessions that align with both task complexity and the brain's attentional capacity. The intensity of neural resource utilization also interacts with mental energy and arousal levels. Complex tasks require greater allocation of attentional networks and more frequent engagement of dopaminergic and noradrenergic pathways that support motivation and vigilance. As these neurotransmitter systems fluctuate during prolonged effort, performance on demanding tasks declines more rapidly compared to simpler tasks. By segmenting complex work into shorter intervals, ideally within the 60 to 90-minute range, and incorporating restorative breaks, individuals can sustain peak performance without overtaxing neural and metabolic resources. This approach also reduces the likelihood of cognitive errors and mental fatigue that are more pronounced during high-complexity activities. Task complexity further interacts with emotional engagement and stress. High-demand tasks often induce higher levels of perceived effort and mental strain, which can accelerate the onset of fatigue and reduce cognitive endurance. Conversely, when complex tasks are emotionally engaging or intrinsically rewarding, they can temporarily boost motivation and resilience, allowing longer periods of focus. However, this effect is limited, and even highly engaging tasks eventually require breaks to maintain optimal cognitive performance. Understanding the interplay between task difficulty, emotional engagement, and neural energy expenditure is crucial for structuring productive work sessions. Personalization of work intervals based on task complexity enhances efficiency and sustainability. Monitoring subjective experiences such as mental effort, fatigue, and frustration, along with objective indicators like error rates or processing speed, allows individuals to adjust the length of focus sessions to match task demands. Complex, unfamiliar, or high-stakes tasks benefit from shorter, highly concentrated work intervals with structured breaks, while repetitive or routine tasks can tolerate longer intervals with minimal performance decline. This adaptive approach ensures that cognitive resources are allocated effectively, maximizing output while minimizing fatigue and stress. Environmental factors and support mechanisms also influence how task complexity affects focus duration. High distraction or high-pressure environments amplify the cognitive load of complex tasks, reducing the effective duration of attention, whereas optimized, low-stress settings extend focus capacity. Similarly, preparation strategies such as task segmentation, prioritization, and pre-planning can reduce perceived complexity and cognitive load, enabling more sustained attention and higher quality performance. Aligning task management with environmental and physiological factors allows individuals to approach complex work in a way that preserves both performance and cognitive energy. By accounting for the cognitive demands and complexity of tasks, individuals can strategically structure work sessions to optimize focus, productivity, and efficiency. Matching work interval length to task difficulty ensures that neural resources are used effectively, performance is maintained at a high level, and mental fatigue is minimized, providing a sustainable framework for SUSTA. Environmental and physiological factors significantly influence the duration and quality of focused work, shaping how effectively attention can be sustained over time. The brain's ability to maintain high-level cognitive performance is not isolated from external conditions. Rather, it is intimately linked to sensory input, ambient conditions, and internal bodily states. Distractions in the environment, such as noise, interruptions, or visual clutter, force the attentional networks of the prefrontal cortex to constantly reorient, increasing cognitive load and accelerating fatigue. Each shift of attention consumes neural resources and neurotransmitters, reducing the overall efficiency of sustained focus. Optimizing the environment by minimizing distractions, controlling sensory input, and creating structured workspaces allows the brain to maintain continuous engagement for longer intervals, making work sessions more productive and less mentally taxing. Lighting and ergonomics also play a critical role in sustaining attention. Natural light exposure supports circadian regulation, influencing alertness and arousal levels, while poor lighting can increase eye strain, fatigue, and stress, reducing the capacity for prolonged focus. Similarly, ergonomically designed workspaces, including supportive seating, proper desk height, and positioning of tools or devices, reduce physical strain that otherwise competes for attentional resources. By aligning the workspace with physiological comfort and sensory optimization, individuals can extend periods of effective focus and reduce the cognitive cost of attending to discomfort or environmental inefficiencies. Internal physiological states, such as hydration, nutrition, and sleep, are equally influential. The prefrontal cortex is highly sensitive to fluctuations in energy availability, and even mild dehydration or low blood glucose levels can impair executive function, attentional control, and working memory. Adequate hydration and balanced nutrition support optimal neural metabolism, neurotransmitter function, and cognitive endurance, allowing individuals to sustain focus for longer intervals. Sleep quality is perhaps the most critical physiological factor. Chronic sleep deprivation or fragmented sleep disrupts prefrontal cortex activity, alters dopamine and norepinephrine regulation, and diminishes attentional resilience. Well-rested individuals exhibit greater capacity for extended focus, more consistent decision-making, and better resistance to distraction, highlighting the necessity of aligning work sessions with internal physiological readiness. Circadian rhythms also determine the optimal timing of focused work sessions. Each individual has natural peaks and troughs of alertness and cognitive energy throughout the day, influenced by circadian biology and ultradian cycles. Scheduling demanding work during periods of peak alertness leverages naturally elevated arousal and attentional capacity, maximizing efficiency and minimizing mental fatigue. Conversely, attempting high-intensity cognitive work during circadian low points increases the likelihood of errors, slower processing, and shorter effective focus intervals. By synchronizing work schedules with internal biological rhythms, individuals can optimize attention, performance, and motivation. Stress and emotional state interact with environmental and physiological factors to modulate focus duration. Elevated stress levels trigger sympathetic nervous system activation, increasing cortisol release, which can impair prefrontal cortex function and reduce attentional control. Supportive environments, stress reduction strategies, and mindfulness practices help maintain arousal within optimal ranges, enabling sustained focus. Even minor adjustments, such as a quiet workspace, pleasant ambient sounds, or brief mindfulness breaks, can significantly enhance cognitive endurance by reducing internal distractions and stress-induced attentional depletion. Temperature and air quality are additional environmental variables that influence focus. Extreme temperatures or poor ventilation can increase discomfort, elevate mental fatigue, and reduce cognitive performance. Maintaining moderate ambient temperature and ensuring adequate airflow support both physiological comfort and neural efficiency. Similarly, background noise should be managed. Some low-level ambient sounds can aid focus, while high-intensity or unpredictable noise disrupts attentional networks and accelerates fatigue. These factors collectively highlight the importance of tailoring the physical and sensory environment to support prolonged cognitive engagement. By integrating environmental and physiological considerations, ranging from workspace ergonomics, lighting, and noise control to hydration, nutrition, sleep, and circadian timing, individuals can create conditions that maximize attentional endurance. Aligning external and internal factors with the natural rhythms and metabolic needs of the brain allows for longer, more efficient, and sustainable periods of focused work. Optimizing these factors ensures that cognitive resources are preserved, mental fatigue is minimized, and performance remains consistent across extended work. By deliberately managing environmental and physiological factors, individuals can create an optimal context for sustained focus and high-level cognitive performance. Attention, motivation, and mental endurance are not solely determined by willpower. They are strongly shaped by workspace conditions, sensory input, internal energy states, and alignment with circadian rhythms. When these variables are optimized through proper lighting, ergonomics, minimal distractions, adequate hydration and nutrition, sufficient sleep, and strategic scheduling, focused work becomes more efficient, less mentally taxing, and more sustainable over time. Integrating these considerations into daily routines ensures that cognitive resources are preserved, mental fatigue is minimized, and each work session achieves maximal productivity and quality output.